Not only does oral delivery of insulin promise to improve the quality of life for up to 40 million people with type 1 diabetes worldwide, it could also mitigate many of the disease's life-threatening side effects that result from patients failing to give themselves required injections. Insulin therapy by injecting just under the skin or delivered by an insulin pump generally keeps the glucose levels of most diabetics in check, but many people fail to adhere to that regime due to pain phobia of needles and the interference with normal activities. The consequences of the resulting poor glycemic control can lead to serious health complications. Finding a way to deliver insulin orally has been elusive. The protein does not fare well when it encounters a stomach's acidic environment and is poorly absorbed out of the intestine. The key to the new approach is to carry insulin in an ionic liquid comprised of choline and uronic acid that is then put inside a capsule with an acidic-resistant enteric coating. The formulation is biocompatible, easy to manufacture, and can be stored for up to 2 months at room temperature without degrading, which is longer than some injectable insulin products currently on the market. Once ingested, insulin must navigate a challenging obstacle course before it can be effectively absorbed into the bloodstream. Our approach is like a Swiss Army knife, where one pill has tools for addressing each of the obstacles that are encountered. By encapsulating the insulin ionic liquid formulation in an enteric coating, the team overcame the first obstacle, resisting breakdown by gastric acids in the gut. The polymer coating dissolves when it reaches a more alkaline environment in the small intestine, where the ionic liquid carrying insulin is released. When a protein molecule such as insulin enters the intestine, there are many enzymes whose function is to degrade the proteins into smaller amino acids. With the ionic liquid, born insulin remains stable. The choline uronic acid formulation also shows to be adept at penetrating two final barriers: the layer of mucus lining the intestine and the tight cell junctions of the intestine wall, through which large molecule drugs such as insulin cannot easily pass. Other researchers have tried various means of surmounting these barriers by re-engineering the insulin molecule, coating it in protective polymers, and introducing additives to inhibit breakdown by enzymes or to enhance absorption. However, no oral insulin delivery product is currently available in the clinic. It has been the holy grail of drug delivery to develop ways to give protein and peptide drugs to insulin by mouth instead of injection. This study shows remarkable results where insulin given by mouth in combination with ionic liquid works about as well as a conventional injection. The implications of this work to medicine could be huge. If the finding can be translated into pills that safely and effectively administer insulin and other peptide drugs to humans. Orally ingested insulin would more closely mimic the way in which a healthy individual's pancreas makes and delivers insulin to the liver, where up to 80% is extracted and the rest is circulated through the bloodstream. It can also mitigate the adverse effects of taking injections over a long period of time. Scientists also noted that the ionic liquid-borne insulin can be prepared in a one-step process that could be readily scaled up for inexpensive industrial production, making the cost of manufacturing the oral formulation easily manageable. Researchers plan to conduct more animal tests of the formulation as well as long-term toxicological and bioavailability studies. The researchers are optimistic that if all goes well, gaining approval for eventual clinical trials in humans will be made easier by the fact that the key ingredients in their ionic liquids, choline and uronic acid, are already considered safe. The Food and Drug Administration has established a daily recommended dose of choline, a vitamin-like essential nutrient, and uronic acid, a chemical that naturally occurs in cardamom and lemongrass, is widely used as a food additive. This is Professor Will's CBMD PhD. Today, Sunday, July 8th, 2018. It is 18:37:40 p.m. Eastern Standard Time. Science News. Test tube artificial neural network recognizes molecular handwriting. Date July 4, 2018. Source: California Institute of Technology. Summary: scientists have developed an artificial neural network out of DNA that can recognize highly complex and noisy molecular information. Researchers at Caltech have developed an artificial neural network made out of DNA that can solve a classic machine learning problem. Correctly identifying handwritten numbers, the work is a significant step in demonstrating the capacity to program artificial intelligence into synthetic biomolecular circuits. Though scientists have only just begun to explore creating artificial intelligence in molecular machines, its potential is already undeniable, similar to how electronic computers and smartphones have made humans more capable than 100 years ago. Artificial molecular machines can make all things made of molecules, perhaps including even paint and bandages, more capable and more responsive to the environment in the 100 years to come. Artificial neural networks are mathematical models inspired by the human brain. Despite being much simplified compared to their biological counterparts, artificial neural networks function like networks of neurons and are capable of processing complex information. The laboratory ultimate goal for this work is to program intelligent behaviors, the ability to compute, make choices, and more, with artificial neural networks made out of DNA. Humans each have over 80 billion neurons in the brain, with which they make highly sophisticated decisions. Smaller animals, such as roundworms, can make simpler decisions using just a few hundred neurons. In this work, we have designed and created biochemical circuits that function like a small network of neurons to classify molecular information substantially more complex than previously possible. Human handwriting can vary widely, and so when a person scrutinizes a scribbled sequence of numbers, the brain performs complex computational tasks in order to identify them. Because it can be difficult even for humans to recognize other sloppy handwriting, identifying handwritten numbers is a common test for programming intelligence into artificial neural networks. These networks must be taught how to recognize numbers, account for variations in handwriting, then compare an unknown number to their so-called memories and decide the number's identity. In other work described in Nature, scientists demonstrate that a neural network made out of carefully designed DNA sequences could carry out prescribed chemical reactions to accurately identify molecular handwriting. Unlike visual handwriting that varies in geometric shape, each example of molecular handwriting does not actually take the shape of a number. Instead, each molecular number is made up of 20 unique DNA strands chosen from 100 molecules, each assigned to represent an individual pixel in any 10 by 10 pattern. These DNA strands are mixed together in a test tube. The lack of geometry is not uncommon in natural molecular signatures, yet still requires sophisticated biological neural networks to identify them. For example, a mixture of unique odor molecules comprises a smell. Given a particular example of molecular handwriting, the DNA neural network can classify it into up to 9 categories, each representing one of the 9 possible handwritten digits, from 1 to 9. Scientists had built a neural network to distinguish between handwritten 6s and 7s. Science tested 36 handwritten numbers, and the test tube neural networks correctly identified all of them. Science system theoretically has the capability of classifying over 12,000 handwritten 6s and 7s. 90% of those numbers taken from a database of handwritten numbers used widely for machine learning into the 2 possibilities. Crucial to this process was encoding a winner-take-all competitive strategy using DNA molecules, developed by science research. In this strategy, a particular type of DNA molecule dubbed the annihilator was used to select a winner when determining the identity of an unknown number. The annihilator forms a complex with one molecule from one competitor and one molecule from a different competitor and reacts to form inert, unreactive species. The annihilator quickly eats up all of the competitor molecules until only a single competitor species remains. The winner competitor is then restored to a high concentration and produces a fluorescent signal indicating the network's decision. Next, scientists built upon the principles of the first DNA neural network to develop one even more complex one that could classify single-digit numbers 1 through 9. When given an unknown number, this smart soup would undergo a series of reactions and output 2 fluorescent signals. For example, green and yellow to represent a 5, or green and red to represent a 9. Researchers plan to develop artificial neural networks that can learn forming memories from examples added to the test tube this way. The same smart soup that can be trained to perform different tasks. Common medical diagnostics detect the presence of a few biomolecules, for example cholesterol or blood glucose, using more sophisticated biomolecular circuits like ours. Diagnostic testing could one day include hundreds of biomolecules with the analysis and response conducted directly in the molecular environment. Today's is Professor Will CBMD PhD. Today is Monday, July 9, 2018. It is 10:05 a.m. Eastern Standard Time. Combining antibiotics changes their effectiveness. Effectiveness of antibiotics altered by combining them with each other. non-antibiotic drugs or even with food additives. Date July 4, 2018. Source European Molecular Biology Laboratory. Summary. The effectiveness of antibiotics can be altered by combining them with each other, non-antibiotic drugs or even with food additives, depending on the bacterial species. Some combinations stop antibiotics from working to their full potential whilst others begin to defeat antibiotic resistance, report researchers. In the first large-scale screening of its kind, scientists profiled almost 3,000 drug combinations on 3 different disease-causing bacteria. The research was led by EMBL. Overcoming antibiotic resistance. Overuse and misuse of antibiotics has led to widespread antibiotic resistance. Specific combinations of drugs can help in fighting multi-drug resistant bacterial infections, but they are largely unexplored and rarely used in clinics. That is why in the current paper, the team systematically studied the effect of antibiotics paired with each other, as well as with other drugs and food additives in different species. Whilst many of the investigated drug combinations lessened the antibiotics effect, there were over 500 drug combinations which improved antibiotic outcome. A selection of these positive pairings was also tested in multi-drug resistant bacteria, isolated from infected hospital patients, and were found to improve antibiotic effects. Critical vanillin. When vanillin, the compound that gives vanilla its distinctive taste, was paired with one particular antibiotic known as spectinomycin, it helped the antibiotic to enter bacterial cells and inhibit their growth. Spectinomycin was originally developed in the early 1960s for treating gonorrhea, but is rarely used nowadays due to the bacterial resistance that was developed against it. However, in combination with vanillin, it could become clinically relevant once again and used for other disease-causing microbes. Of the combinations tested, this was one of the most effective and promising synergies we identified. Pairings such as this could extend the arsenal of weapons in the war against antibiotic resistance. Curiously, however, vanillin lessened the effect of many other types of antibiotics. The paper showed that vanillin works in a similar way to aspirin to decrease the activity of many antibiotics. Although its effects in human cells have not been tested, they are most likely different. According to research, combinations of drugs that decrease the effect of antibiotics could also be beneficial to human health. Antibiotics can lead to collateral damage and side effects because they target healthy bacteria as well. But the effects of these drug combinations are highly selective and often affect a few bacterial species. In the future, we could use drug combinations to selectively prevent the harmful effects of antibiotics on healthy bacteria. This would also decrease antibiotic resistance development, as healthy bacteria would not be put under pressure to evolve antibiotic resistance, which can later be transferred to dangerous bacteria. General principles. This research is the first large-scale screening of drug combinations across different bacterial species in the lab. The compounds used have already been approved for safe use in humans, but investigations in mice and clinical studies are still required to test the effectiveness of particular drug combinations in humans. In addition to identifying novel drug combinations, the size of this investigation allowed the scientists to understand some of the general principles behind drug-drug interactions. This will allow more rational selection of drug pairs in the future and may be broadly applicable to other therapeutical areas. Today's is Professor Will, CBMD, PhD. Today's Monday, July 9, 2018 is 10:26:32 seconds a.m. Eastern Standard Time. Science News. Fragment of impacting asteroid recovered in Botswana. Date July 6, 2018. Source University of Helsinki. Summary. On Saturday, June 23, 2018, a team of experts from Botswana, South Africa, Finland, and the United States of America recovered a fresh meteorite in Botswana's central Kalahari Game Reserve. The meteorite is one of the fragments of asteroid 2018 LA, which collided with Earth on June 2, 2018 and turned into a meteor fireball that detonated over Botswana a few seconds after entering the atmosphere. The incident was witnessed by a number of spectators in Botswana and neighboring countries and was captured on numerous security cameras. Asteroid 2018 LA was detected in space 8 hours before hitting Earth. It was detected by Catalini Sky Survey, operated by the University of Arizona and sponsored by NASA as part of its planetary defense mission. This is the third line in history that an asteroid inbound to hit Earth was detected early and only the second time that fragments were recovered. After disruption, the asteroid fragments were blown by wind while falling down, scattering over a wide area. Calculations of the landing area were done independently by researchers. Today's is Professor Will, CBMD, PhD. Today's Monday, July 9, 2018, and it's 10:41:45 seconds a.m. Eastern Standard Time. Science News. Semiconductor quantum transistor opens the door for photon-based computing. Date July 5, 2018. Source University of Maryland. Summary. Researchers have demonstrated the first single photon transistor using a semiconductor chip. Transistors are tiny switches that form a bedrock of modern computing. Billions of them route electrical signals around inside a smartphone, for instance. Quantum computers will need analogous hardware to manipulate quantum information, but the design constraints for this new technology are stringent, and today's most advanced processors can't be reproposed as quantum devices. That's because quantum information carriers, dubbed qubits, had to follow different rules laid out by quantum physics. Scientists can use many kinds of quantum particles as qubits, even the photons that make up light. Photons have added appeal because they can swiftly shuffle information over long distances, and they are compatible with fabricated chips. However, making a quantum transistor triggered by light has been challenging because it requires that the photons interact with each other, something that doesn't ordinarily happen on its own. Using our transistor, we should be able to perform quantum gates between photons. Software running on quantum computer would use a series of such operations to attain exponential speed up for certain computational problems. The photonic chip is made from a semiconductor with numerous holes in it, making it appear much like a honeycomb. Light entering the chip bounces around and gets trapped by the hole pattern. A small crystal called a quantum dot sits inside the area where the light intensity is strongest. Analogous to conventional computer memory, the dot stores information about photons as they enter the device. The dot can effectively tap into the memory to mediate photon interactions, meaning that the actions of one photon affect others that later arrive at the chip. In a single photon transistor, the quantum dot memory must persist long enough to interact with each photonic qubit. This allows a single photon to switch a bigger stream of photons, which is essential for our device to be considered a transistor. To test that the chip operated like a transistor, the researchers examined how the device responded to weak light pulses that usually contained only one photon. In a normal environment, such dim light might barely register. However, in this device, a single photon gets trapped for a long time, registering its presence in the nearby dot. The team observed that a single photon could, by interacting with a dot, control the transmission of a second light pulse through the device. The first light pulse acts like a key opening the door for the second photon to enter the chip. If the first pulse didn't contain any photons, the dot blocked subsequent photons from getting through. This behavior is similar to the conventional transistor, where a small voltage controls the passage of current through its terminals. Here the researchers successfully replaced the voltage with a single photon and demonstrated that their quantum transistor could switch a light pulse containing around 30 photons before the quantum dot's memory ran out. Good day, this is Professor Will CBMD PhD. Today's Sunday, June 24th, 2018. It is 7:47 seconds a.m. Eastern Standard Time. Science News. Psychiatric disorders share an underlying genetic basis. Psychiatric disorders may have important molecular similarities and are not reflected in current diagnostic categories. Date June 21st, 2018. Source Broad Institute of MIT and Harvard. Summary. Researchers explored the genetic connections between brain disorders on a scale far eclipsing previous work on the subject. The team determined that psychiatric disorders share many genetic variants, while neurological disorders such as Parkinson's or Alzheimer's appear more distinct. The results indicate that psychiatric disorders likely have important similarities at a molecular level which current diagnostic categories do not reflect. Psychiatric disorders such as schizophrenia and bipolar disorder often run in families. In a new international collaboration, researchers explored the genetic connections between these and other disorders of the brain at a scale that far eclipses previous work on the subject. The team determined that psychiatric disorders share many genetic variants, while neurological disorders such as Parkinson's or Alzheimer's appear more distinct. Published today in Science, the study takes the broadest look at how genetic variants relate to brain disorders. The results indicate that psychiatric disorders likely have important similarities at a molecular level which current diagnostic categories do not reflect. This work is starting to reshape how we think about disorders of the brain. If we can uncover the genetic influences and patterns of overlap between different disorders, then we might be able to better understand the root cause of these conditions and potentially identify specific mechanisms appropriate for tailored treatments. Exploring these biological connections is challenging. The brain is a tricky organ to study directly, difficult to scan in detail or ethically biopsy. And because brain disorders often co-occur, it is hard to untangle when one might be affecting the development of another. To examine the biological overlap between these disorders, researchers must rely on genetics. For the current study, international consortia pooled their data to examine the genetic patterns across 25 psychiatric and neurological diseases. Because each genetic variant only contributes a tiny percentage of the risk for developing a given disorder. The analysis required huge sample sizes to separate reliable signals from noise. The researchers measured the amount of genetic overlap across the disorders using genome-wide association studies (GWAS) of 265,218 patients and 643 controls. They also examined the relationships between disorders and 17 physical or cognitive measures, such as years of education from 1,191,588 individuals. The dataset ultimately included all GWAS consortia studying common brain disorders that the team could identify with sufficient sample sizes. The final results indicated widespread genetic overlap across different types of psychiatric disorders, particularly between attention deficit hyperactive disorder, ADHD, bipolar disorder, major depressive disorder, and schizophrenia. The data also indicated strong overlap between anorexia nervosa and obsessive compulsive disorder, OCD, as well as between OCD and Tourette syndrome. In contrast, neurological disorders such as Parkinson's and multiple sclerosis appeared more distinct from one another and from the psychiatric disorders, except for migraine, which was genetically correlated to ADHD, major depressive disorders, and Tourette syndrome. According to the researchers, the high degree of genetic correlation among the psychiatric disorders suggests that current clinical categories do not accurately reflect the underlying biology. The tradition of drawing these sharp lines when patients are diagnosed probably doesn't follow the reality, where mechanisms in the brain might cause overlapping symptoms. As a hypothetical example, a single mechanism regulating concentration could drive both inattentive behavior in ADHD and diminished executive function in schizophrenia. Further exploration of these genetic connections could help define new clinical phenotypes and inform treatment development and selection for patients. Additionally, within the cognitive measures, the researchers were surprised to note that genetic factors predisposing individuals to certain psychiatric disorders, namely anorexia, autism, bipolar, and OCD, were significantly correlated with factors associated with higher childhood cognitive measures, including more years of education and college attainment. Neurological disorders, however, particularly Alzheimer's and stroke, were negatively correlated with those same cognitive measures. We were surprised that genetic factors of some neurological diseases normally associated with the elderly were negatively linked to genetic factors affecting early cognitive measures. It was also surprising that the genetic factors related to many psychiatric disorders were positively correlated with educational attainment. We'll need more work and even larger sample sizes to understand these concentrations. The consortia have made their GWAS data accessible online, either free available for download or by application. They plan to examine additional traits and genetic variants to explore these patterns further, aiming to discover the relevant mechanisms and pathways that underlie and potentially link these disorders. Good day, this is Professor Will C. BMD, PhD. Today, Sunday, June 24, 2018. It is 8:27:49 seconds a.m. Eastern Standard Time. Einstein proved right in another galaxy. Date, June 21st, 2018. Source, University of Portsmouth. Summary: astronomers have made the most precise test of gravity outside our own solar system. By combining data taken with NASA Hubble Space Telescope and European Southern Observatory Very Large Telescope, the researchers show that gravity in this galaxy behaves as predicted by Albert Einstein's general theory of relativity, confirming the theory validity on galactic scales. An international team of astronomers have made the most precise test of gravity outside our own solar system. By combining data taken with NASA Hubble Space Telescope and the European Southern Observatory Very Large Telescope, the researchers show that gravity in the galaxy behaves as predicted by Albert Einstein's general theory of relativity, confirming the theory validity of galactic scales. In 1915, Albert Einstein proposed his general theory of relativity, GR, to explain how gravity works. Since then, GR has passed a series of high-precision tests within the solar system, but there have been no precise tests of GR on large astronomical scales. It has been known since 1929 that the universe is expanding, but in 1998, two teams of astronomers showed that the universe is expanding faster now than it was in the past. This surprising discovery, which won the Nobel Prize in 2011, cannot be explained unless the universe is mostly made of exotic component called dark energy. However, the interpretation relies on GR being the correct theory of gravity on cosmological scales. Testing the long-distance properties of gravity is important to validate our cosmological model. A team of astronomers led by Dr. Thomas Collett, the Institute of Cosmology, Gravitation, and University of Portsmouth, used a nearby galaxy as a gravitational lens to make a precise test of gravity on astronomical length scale. General relativity predicts that massive objects deform space-time. That means that when the light passes near another galaxy, the light path is deflected. If two galaxies are aligned along our line of sight, this can give rise to the phenomenon called strong gravitational lensing, where we see multiple images of the background galaxy. If we know the mass of the foreground galaxy, then the amount of separation between the multiple images tells us if general relativity is the correct theory of gravity of galactic scales. A few hundred strong gravitational lenses are known, but most are too distant to precisely measure their mass, so they can't be used to accurately test GR. However, the galaxy ESO 325 G004 is amongst the closest lenses at 500 million light-years from Earth. We used data from the Very Large Telescope in Chile to measure how fast the stars were moving in E325. This let us infer how much mass there must be in E325 to hold these stars in orbit. We then compared the mass to the strong lensing image separation that we observed with the Hubble Space Telescope, and the result was just what GR predicts with 9% precision. This is the most precise extrasolar test of GR to date from just one galaxy. The universe is an amazing place, providing such lenses which we can then use as our laboratories. This is Professor Will, CBMD, PhD. Today, Sunday, June 24, 2018. It is 9:54:20 a.m. Eastern Standard Time. Science News. Nearly 80 exoplanet candidates identified in record time. Search considered successful dress rehearsal for exoplanet hunter tests. Date, June 21st, 2018. Source, Massachusetts Institute of Technology. Summary, scientists have analyzed data from K2, the follow-up mission to NASA Kepler Space Telescope, and have discovered a trove of possible exoplanets amid some 50,000 stars. The scientists report the discovery of nearly 80 new planetary candidates, including a particular standout, a likely planet that orbits the star HD 73344, which would be the brightest planet host ever discovered by the K2 mission. In a paper that appeared online today in the Astronomical Journal, the scientists report the discovery of nearly 80 new planetary candidates, including a particular standout, a likely planet that orbits the star HD 73344, which would be the brightest planet host ever discovered by the K2 mission. The planet appears to orbit HD 73344 every 15 days, and based on the amount of light that it blocks each time it passes in front of the star, scientists estimate that the planet is about 2.5 times the size of the Earth and 10 times as massive. It is also likely incredibly hot, with temperature somewhere in the range of 1,200 to 1,300 degrees Celsius, or around 2,000 degrees Fahrenheit, about the temperature of lava from an erupting volcano. The planet lies at a relatively close distance of 35 parsecs, or about 114 light-years from Earth. Given its proximity and the fact that it orbits a very bright star, scientists believe the planet is an ideal candidate for follow-up studies to determine its atmospheric composition and other characteristics. The new analysis is also noteworthy for the speed with which it was performed. The researchers were able to use existing tools developed at MIT to rapidly search through graphs of light intensity called light curves from each of the 50,000 stars that K2 monitored in its two recent observing campaigns. They quickly identified the planetary candidates and released the information to the astronomy community just weeks after the K2 mission made the spacecraft's raw data available. A typical analysis of this kind takes between several months and a year. Crossfield says such a fast planet search enables astronomers to follow up with ground-based telescopes much sooner than they otherwise would, giving them a chance to catch a glimpse of planetary candidates before the Earth passes by that particular patch of sky on its way around the Sun. Such speed will also be a necessity when scientists start receiving data from NASA Transiting Exoplanet Survey satellite tests, which is designed to monitor nearby stars in 30-day swaths and will ultimately cover nearly the entire sky. When the test data come down, there will be a few months before all the stars that tests looked at for that month set for the year. If we get candidates out quickly to the community, everyone can start immediately observing systems discovered by tests and doing a lot of great planetary science, so this analysis was really a dress rehearsal for tests. Speed dips. The team analyzed data from K2 16 and 17 observing campaigns known as C16 and C17. During each campaign, K2 observes one patch of the sky for 80 days. The telescope is on an orbit that trails the Earth as it travels around the sun. For most other campaigns, K2 has been in a rear-facing orientation in which the telescope observes those stars are essentially in its rear view mirror. Since the telescope travels behind the Earth, those stars that it observes are typically not observable by scientists until the planet circles back around the sun to that particular patch of sky, nearly a year later. Thus, for rear-facing campaigns, Crossfire says there has been little motivation to analyze K2 data quickly. The C16 and C17 campaigns, on the other hand, were forward-facing. K2 observed those stars that were in front of the telescope and within Earth's field of view, at least for the next several months. Crossfield and their colleagues an opportunity to speed up the usual analysis of K2 data to give astronomers a chance to quickly observe planetary candidates before Earth passed them by. During C16, K2 observed 20,647 stars over 80 days between December 7, 2017 and February 25, 2018. On February 28th, the mission released the data in the form of pixel-level images to the astronomy community. Crossfield immediately began to sift through the data using algorithms developed at MIT to winnow down the field from 20,000 some stars to 1,000 stars of interest. The team then worked around the clock looking through these 1,000 stars by eye for signs of transits or periodic dips in starlight that could signal a passing planet. In the end, they discovered 30 highest quality planet candidates whose periodic signatures are especially likely to be caused by transiting planets. Our experience with 4 years of K2 data leads us to believe that most of these are indeed real planets ready to be confirmed or statistically validated. The researchers write in their paper. They also identified a similar number of planet candidates in the recent C17 analysis. In addition to these planetary candidates, the group also picks out hundreds of periodic signals that could be signatures of astrophysical phenomena such as pulsating or rotating stars, and at least one supernova in another galaxy. Stars in space. While the nature of the star doesn't typically change over the course of a year, Crossfield says the sooner researchers can follow up on a possible planetary transit, the better chance there is of confirming that a planet actually exists. You want to observe candidates again relatively soon, so you don't lose the transit altogether. Crossfield says you might be able to say, I know there is a planet around the star, but I am no longer at all certain when the transit will happen. That's another motivation for following these things up more quickly. Since the team released its results, astronomers have validated 4 of the candidates as definite exoplanets. They have been observing other candidates that the study identified, including the possible planet orbiting HD 73344. Crossfield says the brightness of the star combined with the speed with which its planetary candidate was identified can help astronomers quickly zero in on even more specific features of the system. We found one of the most exciting planets that K2 has found in its entire mission, and we did it more rapidly than any effort has done before. Crossfield says, this is showing the path forward for how the test mission is going to do the same thing in spades, all over the entire sky, for the next several years. Today's is Professor Will, CBMD, PhD. Today's Sunday, June 24, 2018. It is 12:27 p.m. Eastern Standard Time. Science News. Stealth material hides hotspots from infrared eyes. Date, June 22, 2018. Source, University of Wisconsin Medicine. Summary: infrared cameras are the heat-sensing eyes that help drones find their targets even in the dead of night or through heavy fog. Hiding from such detectors could become much easier, thanks to a new cloaking material that renders objects and people practically invisible. Infrared cameras are the heat-sensing eyes that help drones find their targets even in the dead of night or through heavy fog. Hiding from such detectors could become much easier, thanks to a new cloaking material that renders objects and people practically invisible. What we have shown is the ultra-thin stealth sheet. Right now, what people have is much heavier metal armor or thermal blankets. Warm objects like human bodies or tank engines emit heat as infrared light. The new stealth sheet, described this week in the Research Journal of Advanced Engineering Materials, offers substantial improvements over the heat masking technology. It is a matter of weight, the cost, and ease of use. Less than 1 millimeter thick, the sheet absorbs approximately 94% of the infrared light it encounters. Trapping so much light means that warm objects beneath the cloaking material become almost completely invisible to infrared detectors. Importantly, this stealth material can strongly absorb light in the so-called mid and long wavelength infrared range, the type of light emitted by objects at approximately human body temperature. By incorporating electronic heating elements into the stealth sheet, the researchers have also created a high-tech disguise for tracking infrared cameras. You can intentionally deceive an infrared detector by presenting a false heat signature. It could conceal a tank by presenting what looks like a simple highway guardrail. To trap infrared light, colleagues turned to a unique material called black silicon, which is commonly incorporated into solar cells. Black silicon absorbs light because it consists of millions of microscopic needles called nanowires, all pointing upward like a densely packed forest. Incoming lights reflect back and forth between the vertical spirals, bouncing around within the material instead of escaping. Although black silicon has long been known to absorb visible light, colleagues were the first to see the material potential for trapping infrared. They boosted its absorptive properties by tweaking the method through which they created their material. We didn't completely reinvent the whole process, but we did extend the process to much taller nanowires. They make those nanowires by using tiny particles of silver to help etch down into a thin layer of solid silicon, which results in a thicket of tall needles. Both the nanowires and the silver particles contribute to absorbing infrared light. The researchers' black silicon also has a flexible backing interspersed with small air channels. Those air channels prevent the stealth sheet from heating up too quickly as it absorbs infrared light. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, June 24, 2018. It is 15:10:37 seconds, p.m. Eastern Standard Time. Science News. Putting the brakes on metastatic cancer. Medical research finds 11 genes responsible for the spread of cancer. Date June 20, 2018. Source University of Alberta. Faculty of Medicine and Dentistry. Summary. A groundbreaking discovery has identified previously unknown therapeutic targets that could be key to preventing the spread of cancer. Researchers found that by inhibiting several newly identified gene targets, they could block more than 99.5% of cancer metastasis in living cells. The newly study, published in Nature Communications, this team found that by inhibiting several newly identified gene targets, they could block more than 99.5% of cancer metastasis in living cells. Metastasis kills 90% of all patients with cancer. With this study, we have discovered 11 new ways to potentially end metastasis. In the study, the team used a unique platform and created a shell-less avian embryo to visualize the growth and spread of cancer cells in real time. The researchers used a molecular tool called a knockout library to insert short hairpin RNA shRNA vectors into cancer cells, which bound to specific genes in the cells and stopped them from activating. They then inserted those cancer cells into the embryos and observed as they formed clusters of cancer, identifying which ones showed properties of being non-metastatic. Good day, this is Professor Will, CBMD, PhD. Today is Tuesday, June 26, 2018. 12:59:49 seconds, p.m. Eastern Standard Time. Delivering insulin in a pill. Technique could replace daily injections for diabetics. Date June 25, 2018. Source Harvard John and Paulson School of Engineering and Applied Sciences. Summary. Researchers have developed an oral delivery method that could dramatically transform the way in which diabetics keep their blood sugars level in check. Given the choice of taking a pill or injecting oneself with a needle, most of us would opt to regulate a chronic health condition by swallowing a pill. But for millions of people living with type 1 diabetes, a painful needle prick once a day is the only option for delivering the insulin that their bodies cannot produce on their own. Now, researchers at the Harvard John A. Paulson School of Engineering and Applied Sciences, SEAS, have developed an oral delivery method that could dramatically transform the way in which diabetics keep their blood sugars level in check.